start by saying that osteopontin is a truly multifunctional protein. It is expressed in, so to say, all tissues, and it has a lot of different functions in these different tissues. It's involved in bone metabolism, it's involved in immunology, it's involved in interaction with cells, transport, and everything else. If we look particularly on osteopontin from milk, which is also called Lactopontin, it is special in the way that it is by far the highest concentration of osteopontin is found in milk. It is hundreds of times higher than, for instance, in plasma or in any other tissue that has been analyzed. Also, the specific structure of lactopontin or milk osteopontin differs from other forms of osteopontin in that it's very, very strongly or heavily modified by post-translational modifications, that is phosphorylations and glycosylations. Other forms of osteopontins, for instance in bone or in urine or in plasma, can also be modified, but to a much lesser extent than the milk form. The milk form can contain as much as 34 different sites of phosphorylation and 3 to 5 O-glycosylations, and that's much higher than any other forms of osteopontin. The modifications of osteopontin is actually what makes the protein unique. There's only one gene for osteopontin. It's encoded on chromosome 4, the human chromosome 4, but there are hundreds, if not thousands, of different isoforms, and these isoforms, they differ in the degree of post-translational modification. As said, the milk protein contains a lot of phosphates and also a lot of glycosylations. These modifications are important for the function of the protein. These phosphorylations can interact with, for instance, calcium or other metal ions like zinc or iron, and they can thereby, it can transport these ions to sites of uptake, for instance, in the intestine. These phosphorylations has also been shown to be important for the interaction with integrins. Osteopontin interact with integrins, that is cell receptors, and by this interaction osteopontin can mediate signals and initiate signaling cascades that can make the protein have effects everywhere in the body, either via interaction with these integrins in the intestine, starting the cascade, or actually also it has been shown in many studies that parts of osteopontin can be taken up over the intestinal barrier, going into circulation, and thereby it can actually reach different parts of the body and might have a function there. Especially about the glycosylations, the position of the glycosylations in milk osteopontin or lactopontin, they are placed right next to very important site in osteopontin, which is a site of integrin binding. There are two sequences in osteopontin that can bind these cell receptors, the integrins, and the glycosylations, they are placed very close to these sites. So it's actually hypothesized that these glycosylations protect the integrin binding sites from gastric digestion. We have shown in the lab that if we take osteopontin and we remove the glycosylations of osteopontin, or if we use recombinant osteopontin, then the protein is completely cleaved by pepsin from the gastric, from the stomach. If we use the native lactopontin, the milk osteopontin form, and subject that to the same treatment with gastric aspirates, the sites are not cleaved. So the phosphorylations, the glycosylations kind of protect the important integrin binding sites. So that could be a mechanism by how osteopontin can affect its physiological function or effects after being ingested by a consumer or an infant. That is an interesting question, and it's something that many people are interested in these days. There's a lot of stuff going on about precision fermentation of osteopontin, where many different universities and companies are interested in expressing osteopontin in a microbe and then purifying it. And there are some problems about that, because osteopontin is very heavily modified by post-translation modification, And it's very difficult, I would actually claim it's impossible to do the expression of recombinant osteopontin with the same modification as the one that we find in milk. That is due to the number of phosphorylations. There are between 20 and 35 phosphorylations on milk osteopontin, and also the placement of these phosphorylations in the sequence. They have to be attached to the correct residues to have the correct function. And that's something that is not really doable by recombinant techniques that I know of. Also, then there are the glycosylations that I talked about earlier. These demands a completely different battery of glycosidases, that is enzymes that attach these sugar chains to the molecule. Also, these have to be expressed and be present in this system where you express the recombinant osteopontin. So in conclusion, to make a recombinant form that looks exactly like the milk osteopontin form, that's not really possible. You can do something that comes part of the way, and you might have some of the functions, but we will never have the full battery of function. There was an interesting study years ago where the recombinant osteopontin was compared to milk osteopontin in a mice model study where a mice was induced with dextran sulfate sodium to induce colitis. And in this mice model, then they were treated for 7 days after the induction of the colitis with either recombinant osteopontin with the control water or saline, and then with milk osteopontin. And it was very, very clear to see that after 7 days, the mice that received the milk osteopontin had significantly less symptoms of colitis compared to the ones that received the recombinant form of osteopontin. And that is probably due to some kind of interaction with integrins or receptors that needs the presence of the phosphates in the protein. Osteopontin is very modified post-translationally. And there must be a reason for that. Nature do not invest a lot of energy in adding on phosphates and glycosylations onto a protein if they are there for no reason. So if you produce a recombinant protein that do not have any of these modifications, you simply don't have the same protein. You need these modifications. They are there for a reason, because there are some people that mean that these modifications, they are not that important because it's just a protein that's made to be eaten. But if it was just a protein made to be eaten, then why invest so much energy in adding so many modifications? Because it is quite expensive. It consumes a lot of energy for the organism that produces these proteins to post-translation modify them to such a high degree. So I think that evolution would have wiped out all of these modifications if they had no function. Instead, it actually has put on the highest number of modifications. I think it's one of the most phosphorylated proteins that's ever been described. So it must actually have some kind of function. There are many different things that you can imagine, and you can see that osteopontin can do when included in infant formula. One of the obvious things, if you look at the structure of osteopontin, is that it carries a lot of phosphates, and these phosphates can interact with calcium. So it might be so that osteopontin is a transport of calcium in the milk. But then again, you could say, okay, there's a lot of casein in milk also, and it's present in much higher concentrations, so probably osteopontin is not, the main function of osteopontin is not to carry calcium. Then we have shown in a study years ago that osteopontin might function as an opsonin, that is a marker of bacteria or things present in the milk that are not supposed to be there. They can be marked for removal by macrophages. Osteopontin has been shown to bind to bacteria and also to induce some kind of chemotaxis from macrophages, which then move towards the marked bacteria, and they have also been shown to phagocytize those, to eat them and remove them from the milk. A third function of osteopontin in milk could be that it, via the integrin binding sites present in the center of osteopontin, it can bind to cells and thereby elicit a signal. It has been shown that osteopontin, for instance, can can bind to lamina propria cells in the intestine and thereby induce secretion of interleukin 12, which is an indicator of activation of the innate immune response. So osteopontin might be involved in directing the immune responses in the infants. There's a number of studies that have investigated the concentration and the level of osteopontin in mother's milk. And we see some kind of difference depending on the geographical origin of these mothers. There are some hypotheses that it might be due to genetic inheritance, or it might also be due to diet or something else. I think it could be very interesting to dig a bit deeper into that and have kind of a large cohort that were very well monitored over a longer period of time, so that we could try to deduct what is the reason for having such a high osteopontin or lactopontin concentration in the milk. And then the big question then, if you could also follow the children in years after they have been nursed by these mothers to see whether there could be found any correlations between the ones that had received osteopontin in high concentrations compared to those who had received it in lower concentration. It could be in the development of allergies or infections or inflammations or eczemas or whatever. We don't really know. We can see that osteopontin is involved in a lot of stuff. But if we could actually correlate high osteopontin in mother's milk with some kind of physiological outcome, that could be very interesting.